tonight's speaker brings those two themes together. Stephen Palumbi is the Jane and Marshall Steele Professor of Marine Sciences at Stanford, and also the Harold A. Miller Director of the Hopkins Marine Station. His work focuses on both basic evolutionary questions, but also on the challenges of learning how we can best protect ocean diversity in the face of climate change and other things that affect it. He's also a popular author who, among other books, has written a book called The Extreme Life of the Sea, which is echoed in the title of tonight's talk. A little disappointing that it doesn't include Godzilla or mermaids or krakens, but it has a great deal of information about the fascinating denizens of the deep and what their adaptations tell us about how they may be able to respond to climate change, which will be his theme tonight. Some of his other popular books include The Evolution Explosion, which is about how human beings are forcing evolution upon all of our fellow denizens of the planet and speeding up their evolutionary response. And also The Death and Life of Monterey Bay, which is a hopeful story of an ecosystem and an area being restored and coming back. In addition to being an eminent scientist and a writer and scuba diver, Steve has one other talent. He's also in a band, Sustainable Soul, I understand, together with his daughter. And at the end of the show, or at this webinar, we will play Crab Love. It's a rare opportunity to listen to what's probably the nation's #1 marine ecology themed rock band. So hang around for that after the Q&A. And with that, we are really honored to have Stephen Palumbi. Take it away.
Great. Wow. Thank you. Thank you very, very, very much. And thanks for putting this all together. But also just amazing congratulations for the 50-year run for Ocean Shores. Part of the topic of the death and life of Monterey Bay was just the really important critical role of people in turning around the health and the safety and the ecosystem quality of Monterey and Monterey Bay, and how it really does still matter what people do to help the environment that's around. So thanks for all of that. I spent a lot of time on the Oregon coast. It's a fabulously gorgeous place, and thanks for preserving that. I'm gonna sort of start off with basically a little bit of a view of the ocean that is about how amazing and wonderful it is. So I'm gonna actually maybe be a tiny little bit goofy at the beginning here, because after all, it's summertime now, and we're all feeling a little bit more hopeful. And if you can't just really love and be just incredibly full of wonder about the ocean, then I don't know, that's such the most amazing thing. So I'm going to talk a little bit about just some of the amazing critters that live in the ocean and what we know about them. That's sort of the subject of the extreme life of the sea, and then segue a bit into the climate change world, and then what it is that kind of information can tell us about what to think about climate change effect on the ocean, and what modern science in a lot of parts of the world is trying to do about it. But unfortunately for Philip, that little thing about Godzilla gave me just enough time to add something to this talk, and that's going to be the biggest link I could possibly make to Godzilla, and it's totally within the theme of extreme life of the sea. Part of the book is about how old are species in the sea? How do we tell? And one of the really stunning things that has come out of people's work in the last couple of decades, like Greg Kaye at Moss Landing Marine Lab, is how old some of the species of rockfish are along the coast, like this golden-eyed rockfish. These animals, they could be 2 feet long, 2.5 feet long, but they could be 100 or 120 years old. And knowing that was a big change then in how we would manage those fisheries. But how do we know how long a fish lives? How do we know what its lifespan is? And that's where we begin to get into a little bit of Godzilla, because the way we know about that is by taking advantage of the C14 radioactive isotopes that were blasted into the air by the atomic testing that we did, mostly us, in Bikini Atoll and Aniwetok Atoll of the late 1940s and 1950s. And those open air thermonuclear bombs raised the level of C14, radioactive carbon, in the atmosphere on this graph. That's what's shown starting from like 1955 and on. And what that does is it injects a radioactive signature into the bones of fish that were alive at that time. And a lot of the times we tell how old fish are by counting the bands of something called an otolith, or the ear bone, and that's shown down here in the lower left. And if that ear bone has the radioactive signature that matches what you're seeing on the screen there, then we know that A, that fish was alive in that period of time, and then B, depending upon where that radioactive band is, on the otolith, we can use that to calibrate how old it is. And that's how we know that these rockfish live to 100 or 120 years of age, because of that. So do we know what's the link between that and Godzilla? Well, unfortunately, when we let off the first of our thermonuclear hydrogen bombs on Bikini Atoll, there was a Japanese fishing boat close by, and that Japanese fishing boat got rained down on by the fallout from that first thermonuclear blast, and it limped back home to Japan and caused such a scare because of the radioactivity that it gave birth to the Godzilla legend. And so that's the link between the age of golden-eyed rockfish and Godzilla in this. Now, you can't read about that in The Extreme Life of the Sea. This is a special presentation just for Philip in honor of the 50th birthday there. But what is also in the book, and what we can talk about, is just other ways that fish and marine life are amazing creatures, and how they live their lives and the kinds of abilities they have. For example, there's a chapter in the book about the fastest fish. And how they get that way, what it is that it takes, and we can start with flying fish. For example, flying fish, actually it's kind of a cheat, because flying fish are only fast out of water, not in the water. And it turns out they don't actually fly, they glide. And that has a big role in the way that other critters interact with them in the ocean. So this is a shot of a flying fish with its pectoral fins splayed out the way they are. That's the gliding surface. They don't really have as much muscle on those pectoral fins as, say, birds or bats do, so they can't really flap them. Instead, when they take off and get airborne, it's leaping out of the water and then pushing along with their tail. So this amazing shot shows the lower tail in the water pushing the fish along, and those spread out fins catching the air that takes off and then glides around. Why does it bother to do anything like that at all? And the answer to that is predation. Mahi-mahi in particular have more flying fish in their stomachs than any other fish, and they are a primary predator on these small fish in the tropics. Lots of other fish eat them as well. Well, the end of the story, though, is that the interaction between mahi-mahi and flying fish is complicated, because mahi can swim just about as fast as flying fish can glide. And so you have a situation oftentimes where the flying fish is out of the water, but the predator is underneath tracking it. And because that flying fish is not flying, because the flying fish is gliding, it's coming down as it moves and eventually has to hit the water again. So that's the point where the mahi can strike the flying fish and take it, or miss, in which case it's lost its momentum and the flying fish can jump back out and then continue on its flight. So this fast fish duel is the flying fish and the mahi both being fast for different reasons and in different media. So if flying fish are a little bit of a cheat, because they're out of the water, then what are the real fastest fish? And it's actually a little bit hard to find really good data on that, but you eventually come down to the billfish. Sailfish, marlins are the fastest fish in the ocean. And you can look at that, you can say, yeah, they're fast because they're really muscular and they're streamlined, and they're fast because they are basically trying to catch these small bait fish that move very fast. But along the way, we were trying to basically, I was really wanting to see how fast these fish actually move in the water. They can get up to about 40 miles an hour. And some reports up to 60 or so, but really reliable ones at 40. I just couldn't find a video of those fish swimming at 40 miles an hour. This one is a sailfish going after bait behind a fishing boat in Costa Rica. And this is about 15 miles an hour, and that's the fastest I could find for any clip, any video that actually looked at the sailfish in its native swimming speed, and then nowhere near up to 40 miles an hour. But we really wanted to effectively show this in a set of videos that we produced for the book. So this is what we did. I got a marlin. I got a stuffed marlin, and put it on the roof of my car. And then with the filmmakers at Garthwaite Griffin Films, we basically ran it up and down the Carmel Valley at 40 miles an hour just to see what it looks like at those speeds. So I'm going to show a video of what it looks like at a marlin going by at 40 miles an hour. There, that's what it looks like. And that's why you can't find a video of it, because it is so fast, you just can't get it on screen. So if you want to see it again, for example, that's what it looks like. We kind of gave up looking for video of something going that fast, and dug in a little bit more about what it means to actually move that quickly, and talk about how the biology of the sailfish and the marlins actually leads to the ability to do that. Because here's the other issue about marlins and these billfish. They are not only swimming at 40 miles an hour, they're eating at 40 miles an hour. And in order to be able to see fast enough, because they're swimming very quickly through these baitfish balls, they have to actually heat up their eyeballs. So they have little metabolic cellular heaters behind their eyeballs. This is something discovered by Barbara Block, who's at the Hopkins Marine Station too, and also heaters on their optic lobes in their brains. So by heating up the optic lobes and the eyeball, the visual system of the fish can act and work as fast as the muscles do. But they have to work in tandem in order for the fish not only to swim, but also to be able to eat at 40 miles an hour. So that's what sort of the cool things I think about being able to dig into the extreme life topics. You can actually just take things and then go a little bit deeper and see how it is they're actually working and what that actually means. Moving ahead, there's also parts of this book that talk about different habits, like the deepest and the coldest. And I wanted to sort of stop here in the hottest and talk very briefly about that, because it'll get us into the climate change part of the talk. And the reason for that is that hydrothermal vents, like the ones off the west coast of the U.S., are amazing places. Probably many of you have heard a little bit of those. This is a shot of one of them. They're miles and miles and miles deep. They're places where the tectonic plates of the Earth are moving apart, so there's hot lava right down there, and that superheats the water, causing these what are called black smokers. And all around that are these red tubes, these red tube worms. That are vestimentiferan tube worms. They grow incredibly quickly in a very short period of time, and they were really a huge surprise to biologists, geologists, when they went down into these areas, that there was anything like this kind of amazing life down there. But I want to focus on a relatively unknown one. This is called Alvanella pompeia, and it's called the Pompeii worm, and it lives in the side of these black smoker chimneys. And the front end of the worm, the tentacles here, stick out into the water. It's cold, deep water at about 35 degrees Fahrenheit. But the end of the worm is only an inch away. The end of the worm is stuck into this chimney towards where the black smoke or hot water comes out. And that part of the worm is living at the temperature of hot tea. So this worm goes from ice water to hot tea in its body length of about an inch. And it's a really an amazing ability to be able to live at that temperature. It's the hottest temperature that any metazoan, any kind of worm, any animal lives in. And how it builds its cells, how it how it works, how nerves work, how everything works at that temperature, we haven't the slightest idea. And our biotechnology can't produce that kind of work. So the whole genome of this animal has been sequenced, and people are digging into how the animal actually works, not only at hot temperatures but across this huge range of temperatures. Um, there's a quick setup to trying to think about not just the past, like Philip was talking about, but the future. And if we are all concerned about conservation and the future of the oceans in the next 50 years, then we have to be thinking about not just the extremes that are there now, but future extremes as well, and then how it is that organisms will be able to deal with those. And as many of you undoubtedly know, there's a series of changes coming to the ocean and the whole planet as a result of CO2 going into the atmosphere and the greenhouse gases that cause climate change. And this is just a graph of average ocean temperatures over the last 140 years or so. And compared to now, or compared to some average, that's sort of when ocean shores started in 1971 for the next 30 years or so. So the ocean was cooler than average for the first 100 years, and then gradually hit average, and then has been rocketing above that ever since. So the future of the ocean is warmer, and that has got a lot of people very worried about the future of ecosystems all around. For example, this is the sea surface temperature in 2019 in September compared to the average from 1981 to 2010, and you can see that across a big wide swath of the Pacific, the ocean was much warmer in that particular late summer month than in most other times. And this is just a snapshot. There's just image after image after image after image of this sort of thing. I didn't want to belabor it, but just say, yeah, the ocean is a lot warmer than it has been, and that's affecting organisms in a pretty strong way. But here's a sort of ocean extreme question. If this worm can live at 175 degrees Fahrenheit and has adapted to it, then what's the chance that other species can adapt to warmer conditions? What's the chance that in the future that this kind of warming pattern is something that organisms can adapt to, and adapt to fast enough to maintain ecosystems along the way? So that's kind of the work that we have been doing for the last decade or so, up and down the West Coast here, but also in the tropics, looking at corals. And what I want to talk to you a little bit about is some of that work and our reasons for doing it, but also the way we're going about it. And try at the end to talk a little bit more about what we've been doing on the West Coast about ocean acidification. But in general, if you sort of think about how organisms respond to environmental shift, there's these four ways, MAD, I call it. You can move, acclimate, adapt, or die. And if your sort of conservation ethic is you don't want everything to die, and if you're organisms that don't move very much, then you're basically acclimating, which is changing your physiology to match local conditions, or you're adapting in an evolutionary sense. I do have to say that there's this other way that species would have to adapt to climate change, which is stick your head in the sand and not worry about it. But luckily, over the last decade or so in our country, that's been less and less and less going on. And then we're able to spend more of our time just coming to grips with the future and trying to do something about it. Um, well, movement actually is something that is not only going on as a response to climate change, but is projected to be more and more and more a feature of the kinds of ecosystems that are along our coastline. And this is a little shot of data from a paper a couple years ago by Morley et al., where they looked at the distribution of many fishery species all over the world, really, and they projected the distributions now and how they would change in the future as the oceans warmed up. And in many of these cases, it's because the fish are living along the west coast along a temperature gradient. And as the water warms up, they move to more northern climates, basically keeping their range, their temperature that they experience about the same, but doing that by moving north. And so you can see for example, for the Canary rockfish, it's all up along the west coast and into through through Canada and then into Alaska. And by the time you get to 2080 or 2100, the range also includes a big chunk of the Aleutians and some of that North Pacific. And this is a projected range change just because of the normal way or the way the temperature is imagined to change under certain conditions of CO2 emissions. So this is the movement part of move, move, adapt, acclimate, or die. And it's something that people have been trying to figure out over the last couple of decades with trying to figure out and ecosystem function. Um, what we have been working on more than movement is the adaptation side. What way can evolution maybe help organisms now adapt to future climate in ways that might blunt some of the negative consequences on them? And I sort of emphasize at this point that as far as any of us can tell, we've been working on this. Evolution is a pretty powerful force. It is almost certainly going to be working, but it's almost certainly not going to be enough to solve the problem unless we keep climate change to be fairly mild or moderate. And under those circumstances, we have a decent chance. So I'll spend a bit of time now sort of filling that in. I just wanted to get the take-home message out. And this evolution is actually pretty common and pretty visible in ocean situations and in especially situations where human changes in the environment change what is adaptive or what is high fitness for organisms. So in this case, salmon, this is a data set on salmon populations in Alaska. And these are pink salmon, which come back very regularly at a 2-year cycle. And the gill nets on these streams were catching all the big fish. The small fish were getting through, and as a consequence, inherited growth rates were declining in this population over a period of 20 or 30 or 40 years. So essentially causing the population to evolve a more slow growing lifestyle. You can see it in other kinds of cases. This is a case of a bighorn sheep hunting farm in Canada. Hunters will pay a price to get a trophy. They will wait to get the biggest horned male they can, and as a consequence, over the period of time that this hunting area has been open, the average size of bighorns on males has decreased by 30% as the small horned males live longer, mate more, and have more offspring. So those are just two quick examples of wildlife evolution caused by human action. So what is the chance that human action causing climate change then exerts any evolutionary pressure on organisms? And for this, we're going to jump into coral reefs for a little bit, the area that I work in a lot. We work on kelp forests and coral reefs in parallel. But the work I'm going to tell you about now is corals that we have been studying across the Pacific. And the question is, can climate change cause evolution of corals? Here, what's shown is something called coral bleaching, where high temperatures cause the corals to spit out their symbionts, which are algae that feed them. Their tissue turns white. You can see the skeleton underneath, and so that's why it's called coral bleaching. These corals are not dead, but they're going to be dead pretty soon. And this causes massive damage of coral reefs around the world. But when you look closely, and this is an image from Hawaii, you often see this kind of situation where that one coral is bleached, but the one next to it, same species, is not bleached. And so there may be variation in the population for whether a coral will bleach or not. And if there is, and if it's heritable, then that is the raw material of evolution. And we can use that to kind of predict how evolution might actually be able to work in these settings. We have been doing this work in a number of different places. This is Palau, a country just south of the Philippines and a little bit east in the northern Pacific. We're coming in on a boat towards the Palau International Coral Reef Center with researchers there like Victor Nestor shown on the right, and postdocs and grad students of mine and community members on the right. We have been able to use this as a base of operation to look around this archipelago for corals that are more heat resistant than others. So essentially we're kind of going blind out into the reefs, mapping, sampling corals. We bring them back to the lab. We put them in these sorts of tanks. The tanks here have chillers and heaters and are computer controlled, and they essentially mimic the temperatures that the corals are going to see on a really hot low tide on a bright summer sunny day. And by doing that, we can then ask how much do they bleach and how much do they bleach compared to the other ones around them. So we've been doing this for a couple of years in Palau. This is the sort of result that you can get. There's 5 corals shown here, 5 branches along the top there are each of 5 different corals, and then the same coral is down below. And the ones at the top are just in our control tanks. They're 30 degrees. That's their normal color. The ones down below are essentially been treated at 34 degrees. And you can see the ones that are second from the right is the most heat sensitive, and at 34 degrees, that coral branch is completely white. It's essentially dead. It's highly bleached. Whereas others, like the one on the total left or the one next to it, they're at 34 degrees and they don't really care. They don't bleach at all. They have the same color, the same symbionts, and they're perfectly healthy. So that's the difference that we've been looking at. We've been looking for the corals that are highly heat sensitive and the ones that are highly heat tolerant. And then we were just mapping them. That's the first thing we were trying to do. Let's just get a map of where they are so that we can see how many there are in their distribution. So this is the map that we have generated for Palau so far. And there's 2 things to be sort of come out of this type of map. And one of them is that heat resistant colonies, which are all those red dots, are pretty widespread around the archipelago. And they're pretty common. The other thing, though, is that there's some more common than others. And so you have areas here, like in the, if you see my little cursor, the western clump here. These are it's a clump of warm patch reefs. The water's pretty warm, and there's a lot more incidence of heat-resistant corals there than anywhere else. Up here in the very northern part of the lagoon, the water's cooler, and there's a lower frequency of heat-resistant corals. So they're almost everywhere, but there's some more concentrated in places than others. So that map gives us the ability to, for example, go to these western reefs and say we can collect corals there, find out which ones are heat resistant, and then use them perhaps in restoring heat resistant corals around Palau. But this is where the sort of the biology and the science has to come back in. We know that there's a couple different reasons why an organism like a coral might be heat resistant, and those reasons are they might have acclimated living in a warm reef, Maybe the physiology has just adjusted itself to be able to tolerate warm water, and that's the reason why they're heat resistant. Could be that they have the right symbioses to resist heat. We know there's different kinds of symbionts. Maybe they have a heat resistant one, and maybe they have the right genes, and they would be heat resistant no matter where they are. And that's kind of what we're really after, because acclimation and symbioses, if that's the reason why a coral is heat resistant, those are temporary. They're not permanent. You move the coral, it loses that ability. And so that heat resistance is not a feature of the coral. That heat resistance is a feature of where it was living. So you can't use it for, say, restoration someplace else, because it would lose its heat resistance. Only if it's heat resistant because the coral has the right genes will that be permanent. So that's kind of what we're going for, the permanent heat resistance. So how do we tell what's going on there? How do we actually distinguish these things from one another? And actually, the easiest thing is something that foresters do all the time. It's called a common garden experiment, where you bring organisms, trees, or corals in this case, from different places together, and you plant them in the same place, and you let them grow up. That common area, that common garden, then is the thing that removes the influence of, say, living in different environments. And this is an experiment done by former grad student of mine, Megan Morikawa, 4 different species of corals on this little nursery pad. And the ones outlined in red come from areas of the reef that are pretty warm, and for corals that we've tested and we know are heat resistant. And the coral nubbins that are outlined in blue, same species, they're from parents that we know are heat sensitive, parents on the reef. And this particular 2014 and 15, when a natural bleaching event came through. And so Megan then could use that natural bleaching event to ask, were the corals from the heat resistant parents, did they retain their heat resistance or not? And the means say yes. So heat tolerant parents in red here have offspring on the or nubbins, clones in the nursery that bleach a lot less than the clones, the little coral fragments from heat sensitive parents, which bleach a lot more, about 2 or 3 times more. So in this particular case, there is an inheritance of that heat resistance in these corals in ways that make us think that if we move them to a different place, then they'll retain that, and they will be useful for restoration in other sort of places. So if you're sort of thinking about this from an evolutionary biology point of view, then you got variation among these different corals. Some are heat resistant. It's not just temporary heat resistance, it's permanent. It's probably in the genes of these corals, which would be inherited. And so that has variation and differences, and heritability as all the 3 elements of what you need for evolution, probably. And then maybe that will provide the fuel for future evolution of corals as the climate changes. And we don't know the answer to that. A whole set of models has been done in the last couple of years about what things look like, what populations look like when they're having to evolve in the face of climate change. And these models, in this case, have been applied to corals, but actually the model itself doesn't know it's being applied to corals. It could be fish, it could be sea urchins, it could be almost anything. So in this case, this model just out by Lisa McManus, who's in Hawaii right now, shows that there's just a little bit of inheritable variability. That's V equals 0.01 or 0.1, and that would be the green and the yellow lines. Then the coral populations can continue to evolve and can continue to grow if climate change is not too severe. So what's shown here are two circumstances. One is RCP 8.5, which is the business as usual emissions profile. And under that emissions profile, the ocean is slated to get so warm so quickly that in this model, coral populations simply collapse and can't recover. But if climate change is held to a milder level, in this case RCP 4.5, which is about a little more strong than we would get if we kept to the Paris Accords, then this model suggests coral populations can continue to evolve and can keep up in an adaptive evolutionary framework with what it is that they need to in different settings. And these are the Caribbean, the Southwest Pacific, and the Coral Triangle. So this is just a model, but it suggests some really interesting hope that corals populations, if kept big and they have genetic variation for heat resistance, might in fact be able to help evolve a little bit of the way towards stability in the next century or so. I wanted to do two other things in this talk, but I knew that I didn't want to spend hours and hours and hours lecturing. And the two other things I wanted to talk about were the West Coast and ocean acidification. And so I picked an example which was both of those as a way to show the kinds of experimental work that we can do, and we have done in a collaborative way in kelp forests, to try to actually test the hypothesis that evolution can happen in these settings. Because so far I've just shown the variation is there and the model says that evolution could happen. But we managed to do an evolutionary experiment in kelp forests in the context of ocean acidification. And sort of briefly, and I'm happy to talk about the chemistry a little bit involved if you want. As we know, CO2 is going up in the atmosphere. This is the so-called Keeling curve, showing atmospheric concentrations of CO2 up to about 2017 or so. I mean, as CO2 goes up, a good chunk of it, thank goodness, is absorbed in the ocean. Otherwise, the atmosphere would be even higher in CO2 than it is now. But that CO2 in the ocean dissolves, turns into carbonic acid and creates more acidity in the ocean, driving the pH down to points where it begins to affect a whole series of organisms and their ability to make shells, largely. And those of you who are living along the Oregon coast might have directly experienced this with a sort of crisis over the last decade of oyster farms. Baby oysters essentially are not yet. That's urgent. Baby oysters are particularly susceptible to acidified water, and in particular in oyster farms, when trying to grow up that oyster spat, they were failing to grow and dying en masse. And so it took control of the chemistry of that water that the spat were being grown in order to solve that problem. And the basic problem comes from ocean acidification and the rise of CO2 and the drop of natural pH along the seashore. But other organisms are also faced with these same kind of problems. In fact, all organisms are, especially in their larval phase. So we did an experiment with sea urchins. Now, sea urchins are genetically really pretty well known, especially a couple of years ago. And we had enough knowledge about the various genes involved in calcification and how sea urchins build their skeleton that we could set up an experiment that looked at evolution over a very fast time period, natural selection working in sea urchin populations. So this was done at the Bodega Marine Lab and at Hopkins. We basically spawned a bunch of sea urchins in mixed cultures, raised them up in these settings. Each of these little jars has a different proportion of CO2 in it. And so each of these was growing sea urchin larvae under different conditions, some of which were conditions that were right now, some of the conditions were conditions that are be far, far into the future. And what we ended up being able to do at that point then is to harvest those sea urchin larvae that are shown here, and then pull out the genes that each of these sea urchin larvae have. What we've essentially done is started off with a whole big mixed population with lots of different gene types, and exposed it to a whole series of different environments, essentially selecting for the larvae that can grow best in high acidity, high CO2 conditions. Then, by taking those batches of larvae growing in different conditions, they've been selected to different degrees by these different conditions, and then we can go back and compare the genes that each of these batches have across the entire genome, and ask whether or not natural selection has selected for certain gene variants that were within the original population, and that may then give it the chance to evolve in the future in the wild. We basically can see that evolution happen by doing this kind of experiment. So to make that sort of long story short, Melissa Paspini, who was a grad student at the time, and working with folks at the Bodega Marine Lab, found a whole set of genes that had been significantly under natural selection. That is, their gene frequencies, allele frequencies, and allele types has significantly changed when we expose these larvae to high CO2 conditions. And these genes are just gene names, but they're all involved in the skeleton of the larvae as it's growing. And so we don't know exactly what the mechanisms are that relate to this, but the evidence is that these sea urchin populations have the genetic variability already to be able to adapt to future CO2 conditions, at least partially. And that if we do this experiment in the lab, it's essentially saying, what is the capacity of organisms to adapt to future climate change, and how does that potentially change their trajectory in the future as the climate alters? And basically gets us to this idea of adaptive capacity. And climate adaptation is a phrase that tends to be applied to human communities. How do we adapt to high sea levels, king tides, things like that? But it also applies in an evolutionary framework to all kinds of organisms all over the oceans. And in order to understand the adaptive capacity of populations like kelp or the fish involved, we need to know a couple of different things. Is there enough genetic variation in those populations? Is the population big enough so that it can evolve, it can take a hit from um, climate change? Is the arrival of hotter or more acidified conditions slow enough to give the population enough time over generations to actually change its gene frequencies? Or if climate change happens too quickly, then the evolutionary mechanisms and evolutionary speed get outstripped. Once we kind of had an idea about that, people have begun to think about, can we actually help? Can we move highly, say, heat-resistant corals or heat-resistant kelp or acidification-resistant sea urchins into areas that they'll need to be into the future? Can we do that? Will it help? And then should we do that? Because there's pluses to that, but there's also minuses. There's high risks in some places, and we have to balance the risks against the benefits, and ask the question: should we intervene in ecosystems? Do we know enough to intervene in ecosystems? And then also a really important third question is, what happens if we don't do anything? What's the cost of not doing anything, and in the future? In all of these kinds of things, we have basically been out. We've been trying to balance between what we know about the ocean and the magnificent critters in it, and then what we know about the future, given what we have in climate change. And just sort of my own personal little journey for that is that I had a really hard time writing the end of this book. My co-author is my son, Tony, who wrote a big chunk of all of the central part of the of the book. He's a writer living in Sacramento, and he sort of turned over those last chapters to me and just said, "You have to do these." And I kept writing myself into a corner where the ocean was going to die of climate change, and I just couldn't. I just didn't want that to happen, and I actually. Sort of struggled with what do you do in that circumstance? What do we do to get out of that circumstance? What do we do to keep not only hope alive, but how do we make a plan to actually make things better? And I came up with this sort of this grand bargain that yes, climate change is a really crucial part of the future, and CO2 emissions reductions have to be a really big part of that. But I can't do that. That's up to the financial sectors of our society and the religious sectors and the social civic part. I mean, I can support that in every single way I possibly can. But I have a job too that's important as a conservation biologist, as a conservation citizen, and that's to save as much as possible over the next couple of decades, so that when we get through this century of climate change, there's something to grow back from. That coral reefs and kelp forests and seagrass beds and oyster beds are still in existence after this century, and that they'll grow back naturally when climate basically starts getting better. So that's kind of my mission, my job right now as a biologist, is to find the conditions and find the places and find the mechanisms by which we can do as much as possible to assure that things are still alive in the future. Sometimes that's just protecting them. Sometimes that is intervening in ecosystems. Um, Sometimes it's changing very much the way we use ocean ecosystems. But that's the job, and it meshes with the reduction in emissions that are out there. So that's kind of where I want to leave the talk and sort of start the conversation. I'm thrilled to talk to you all about the beautiful part of the world that you all get to live in. Happy to hear what you think about what we've been doing, and I'll just sort of stop there and open it up for questions.